Now, what is AMD? In our retina, we have some very structured layers. One of the most important structures are the photoreceptors. The photoreceptors help us detect light and essentially see. These photoreceptors are usually separated from blood vessels with a distinct membrane as shown on the screen. Now, what happens during AMD is that these blood vessels tend to grow into the structured photoreceptors. Now, this will lead to immune cell dysfunction and further disturbance of the structure within the retina, and eventually will allow the patients to lose their vision. Now, let's talk a little bit more about my data set. My data set actually contained 13 different samples. This means 13 slices of retina from 13 different patients. Now, as you can see on the screen, the retina is a very structured part of our body, and we were able to group the different cell types into distinct colors. And this is an image of one of our retina slices. What was my main research question this summer? By analyzing retina slices, how can we enrich our understanding of AMD disease progression? Let's start from the beginning. First, we took a sample from our target tissue, and then we isolated the individual cells within this tissue. We popped every individual cell so we could find all of its RNA. For the sake of this example, we're going to use these three cells, cells 1, 2, and 3. Now, we were able to count this RNA and put it into a table. So cell 1 has 3 red RNA, 1 blue RNA, and 2 green RNA. So on and so forth for every cell. Now, based on this table, we can tell that cell 1 has the most amount of red RNA, making cell 1 red. Cell 2 has the most amount of blue RNA, making cell 2 blue. And cell 3 has the most amount of green RNA, making cell 3 green. Now, it's important to recognize different types of RNA in these cells because RNA then becomes protein and essentially determines the function of the cell.